Study of meiosis. Materials required: permanent slides of meiosis and a compound microscope. Procedure: 1. Place the prepared slide on the microscope stage. 2. Using low magnification, locate the dividing cells. 3. Once the dividing cells are identified, adjust to high magnification for detailed observation. Observations: meiosis is a type of cell division in which the number of chromosomes is halved, transitioning from diploid to haploid in the daughter cells such as gametes. Let us go into the details about meiosis. Meiosis occurs in two main phases. 1. Meiosis I. 2. Meiosis II. Each of these phases is further divided into 4 distinct stages. 1. Meiosis I. Prophase I, Metaphase I, Anaphase I, and Telophase I. 2. Meiosis II. Prophase II, Metaphase II, Anaphase II, and Telophase II. Now let us examine the changes that occur during meiosis I. Meiosis I occurs after interphase I and is known as reductive division because the cells are reduced from diploid to haploid. As outlined earlier, this process is comprised of 4 distinct stages. Let us now proceed with a detailed examination of each stage. Let's start with prophase I, which is the longest phase of meiosis I. During this stage, the chromosomes condense and move towards the center of the cell. Prophase I is further divided into 5 distinct subphases: leptotene, zygotene, pachytene, diplotene, and diakinesis. Let us first understand the details of leptotene. Leptotene is the first substage of prophase I. In this phase, the chromatin threads condense and become shorter and thicker. The next stage after leptotene is zygotene. In zygotene, the homologous chromosomes form pairs through a process called synapsis. After the pairing of the homologous chromosomes, they are visible as paired chromatin threads, which are referred to as tetrads or bivalents. After the formation of bivalents, the cells enter the next substage called pachytene. In pachytene, the bivalents exchange the gene segments between the non-sister chromatids of the homologous chromosomes. This process is called the crossing over. The specific location where the exchange of chromosomes or crossing over takes place is called chiasma. Once the genes have been exchanged, the cell enters the next substage called diplotene. In diplotene, the bivalents begin to separate, resulting in the separation of the homologous chromosomes. However, the homologous chromosomes remain connected at the chiasma, which is an interesting aspect of this phase. Next, we move on to the final stage of prophase I, diakinesis. In diakinesis, the homologous chromosomes separate completely and regain their individual identity. Additionally, the nuclear membrane and the nucleolus completely disappear at this stage, and the centrosomes move towards the equator of the cell. Now, let us move ahead with the next stage of meiosis I, which is metaphase I. In this stage, the homologous pairs align on the equator of the cell with a random orientation. This arrangement forms a metaphase plate and is a result of independent assortment. The next stage is anaphase I, where the homologous pairs separate and move towards opposite poles. The separated chromosomes are pulled towards the centrioles at either side of the cell. The microtubules begin to shorten, pulling one chromosome of each homologous pair to the opposite poles in a process known as disjunction. The sister chromatids of each chromosome remain connected. the cell begins to elongate in preparation for cytokinesis. Finally, the cell undergoes the last stage of Meiosis I, known as Telophase I. Meiosis I concludes when the chromosomes of each homologous pair arrive at opposite poles of the cell. The microtubules start to disintegrate and the chromosomes are completely pulled apart. A new nuclear envelope forms around each haploid set of chromosomes. The chromosomes uncoil, reverting to their chromatin form, and cytokinesis occurs, resulting in the formation of two non-identical daughter cells. These daughter cells are haploid and are derived from the diploid parent cell. However, the chromosomes remain attached to their sister chromatids. To separate the sister chromatids, each haploid cell undergoes another round of division. Before this second division, the haploid cells enter a brief resting phase called interkinesis or Interphase II. This is a short phase with no significant activity. After completing this phase, the cell is ready for the second round of meiotic division. The second division is called Meiosis II. Meiosis II may begin with interkinesis or Interphase II. Unlike Interphase I, no S phase occurs in Interphase II as DNA replication has already taken place. Thus, only a G phase occurs. Meiosis II is also referred to as equational division as the cells begin and end as haploid cells. Meiosis II consists of 4 phases similar to Meiosis I but with some differences. The first phase is Prophase II. During this stage, the chromosomes begin to condense, becoming short and compact. The nuclear envelope and nucleolus disintegrate. Centrioles and spindle fibers appear, but no crossing over occurs at this stage. Moving on to the next phase, which is Metaphase II. The chromosomes align at the equatorial plane, forming the metaphase plate. Spindle fibers connect to the kinetochore of each sister chromatid. One sister chromatid faces each pole with their arms divergent. Let us see the next stage, which is Anaphase II. The sister chromatids separate and are now referred to as sister chromosomes. The spindle fibers attached to each chromatid shorten, pulling the sister chromosomes towards opposite poles. Finally, we move into the last stage of Meiosis II, which is Telophase II. The cells divide into two and a new nuclear envelope and nucleolus form around each haploid chromosome set. Meiosis II concludes when the sister chromosomes have reached opposing poles. The spindle fibers disintegrate and the chromosomes recoil, transforming back into chromatin. A nuclear envelope forms around each set of haploid chromosomes. Cytokinesis follows, resulting in the formation of 4 haploid daughter cells in total.